Hey, Hammy here, coming to you with the first of the plant videos for the plant unit. We're going to do 4 chapters, chapter 24, 25, 26, 27, at least in this current edition. That might change down the road. In this first chapter, this first video of 24, we want to cover cells and tissues. We're focusing mainly on flowering plants, the angiosperms. And so this first video, we're looking at some of the cells and tissues that we find. And then in future videos, we'll see, like between monocots and dicots, how these different tissues sort of vary and where we find them in the plant varies. First, just a little review between plant and animal cell, the differences. One of the big ones we learn in Bio 1 is plant cells have cell walls and animal cells do not. Okay. Cell junctions we talked about earlier this year. Cells use plasmodesmata. The animal cells use gap junctions, tight junctions, things like that. Cell division involves a cell plate. Because they have a cell wall, they can't just pinch into two like cinching a belt. So they have to build a new cell wall between them. There's no centrioles. And they have plastids, one of the most famous ones being a chloroplast, where photosynthesis happens. And they usually have very large vacuoles. You'll often see kind of a large central vacuole that's full of water, minerals, some pigments, and stuff like that. We kind of, between an animal cell, plant cell, these are kind of the typical cells as we see them drawn. Plant cells usually more square, rectangular shape, whereas animal cells kind of more round shape. That's sometimes true, that's sometimes not. As we go through tissues today, you'll see that not all plant cells are square or rectangular. And again, just kind of throwing this in here too, don't forget there are the prokaryotic cells, which do not have a DNA in a nucleus or other organelles. Plant cells are eukaryotes. They do have their DNA in a nucleus, and they've got chloroplasts, mitochondria, endoplasmic reticulum, rough and smooth, Golgi apparatus, all that stuff we learned in the cell chapter as well. Another thing before we get into some of the specific tissues are stem cells. Remember, stem cells in an organism are sort of those base cells that are ever dividing and can become a lot of other different cells. They give rise to many other different types of specific specialized cells, or we say differentiated cells. One of the most common ones is what we refer to as the apical meristem. Apical tends to be, and we'll look at this in a later video, kind of at the point or the tip of either the stem or branches or the root, where the plant is growing longer. And so those cells there will give rise to the 3 tissues we're going to talk about here in this video: epidermal or covering tissue, ground or filler tissue, and the vascular tissue that transport water and nutrients. Besides apical meristem, there's another one called intercalary meristem. Instead of being at the ends of the plant, bottom or top, the intercalary tend to be in the stem. And so if it is cut, like this bamboo shoot here, it can actually produce new growth from that stem. Same thing with the grass in our lawns. You mow the grass in our lawns, and it seems to grow from the base up. Okay, so that would be the intercalary meristematic tissue that does that. Another type of stem cell that doesn't add sort of primary growth, as in getting longer, or adding longer as in taller or deeper if you're talking about the roots, but in girth or secondary growth, where the plant would be getting thicker, is vascular cambium. So you can see it labeled here on our little stem. So it would be this tissue right in here, okay? And again, stemmatic tissue can become other types of tissue. So you'll see the 2 vascular tissues here, phloem and xylem, which we'll talk about here in a little bit. It will produce xylem to the inside and phloem to the outside. Okay, so you have vascular cambium that produces xylem on the inside, phloem on the outside. And again, In monocots and dicots, this is a little bit different, but in the next video, we'll look closer at that. There is a third one called cork cambium. Cork cambium is found more in woody plants. This is what would be producing the cork, which eventually becomes the bark. And we'll discuss that in a future video as well. Okay, let's get into some of the actual tissues. First off, number one, we've got epidermal tissue. So just like we think of an epidermis, the covering our skin on our outside, the epidermis of a plant is on the outside surface of the plant, whether that's the stem or the leaves or the root. A lot of times, especially on leaves in dry environments, you can find a waxy cuticle. Waxy cuticle will prevent H2O, water loss. Will also, just like the keratin in our skin and so forth, prevent bacteria and germs and stuff from getting into the plant and making it sick. Sometimes the epidermis can be specialized for root hairs. They're showing you this over here. This little seed, cabbage seedling that's sprouting. They'll get on the root hairs on the outer epidermis of the little root tip that's growing. You get all these fine branching cells or root hairs. Again, obviously, the root hairs are going to increase the surface area for absorption of water and minerals in through the roots. Sometimes stems and leaves can have specialized epidermal cells or hairs called trichomes. If any of you know the burning hazel, the horse nettle or stinging nettle, sometimes along creeks and banks, you rub against it and you get this rash, this burning sensation. Prevents foraging from different creatures. Many plants have hairs as well that prevent insects from feeding on them, aphids and so forth. As farmers, there's a variety of alfalfa we grow with trichomes, hairs on the stems that prevent certain bugs from getting in there that suck out the sap and then causes your hay to die, your alfalfa to die, and it doesn't grow anymore and you don't get much hay crop. So they've developed varieties with hairy stems to help prevent that. Kind of like you see over here, and it prevents the aphids from getting to the stem and feeding. Leaves, we've talked about in the photosynthesis chapter, of having these little two guard cells with the opening called the stomata or the stoma. This is where gas exchange is going to happen in the leaf. Where you're taking in CO2 for photosynthesis and getting rid of the oxygen. Woody plants as well, and I mentioned this cork cambium as well, produce cork. They will have specialized openings called lenticels. Lenticels, if you look at a woody twig sometimes, you see these little, see if I can point to one here, these little, look like little dashes or gashes in the bark. Those are just little openings, again, for gas exchange to happen in the stem. Tissue number 2 is ground tissue. Ground tissue is most abundant tissue we find in the plant. It's usually the least specialized, and it's found all in the plant, all the organs. They have organs just like we would think of like a human body having organs. But instead of liver and stomach and so forth, they have roots, shoots, leaves, flowers. Those would all be considered organs of the plant. You know, when you bite into an apple, okay, the first one, there's 3 ground tissues we're going to talk about. Parenchyma, sclerenchyma, and cholenchyma. Okay, the first one, parenchyma, parenchyma, or I usually just call it parenchyma, is the least specialized. When you bite into an apple, most of that apple juice or that apple meat, okay, that you're eating is the parenchyma cells, parenchyma cells. In photosynthesis chapter, we studied about the leaves, the spongy and palisade mesophyll, where a lot of photosynthesis is happening. Guess what? That is parenchyma cells. These cells can also have the ability to divide and give rise to more specialized cells. If you've ever really wanted a plant from somebody, so they take a cutting of a leaf or stem, soak it in water, and they start to grow these little root hairs on them and grow these roots, and you can actually take it and transplant it. So you can actually clone the plant. That comes from the ability of the parenchyma cells. Ground tissue number 2 is Sclerenchyma or Sclerenchyma. I usually again say Sclerenchyma. Sclerenchyma have thicker primary walls, usually in the corners. So if you compare it to the last slide or the picture in your book or on your plant posters, usually, so if this is our plant cell here, you're going to see usually at the corners here, it's going to be thicker. Okay, this is usually for support. Especially immature growing regions of the plant. Here you can see a celery, and on the outside of the celery, it collects the Sclerenchyma out here along the just underneath the epidermis. So if you have ever eaten celery, you know when you take that celery stalk and break it, you kind of get those long strands. Okay, those strands are strands of Sclerenchyma that are providing support to that celery stalk that's growing up off of that base up into the air. Okay, ground tissue number 3. Tissue number 3 is Sclerenchyma. They are even thicker secondary walls as well, usually impregnated with lignin. Lignin is used for strength and support. And the Sclerenchyma cells, and you can see from the picture over here, the microscopic picture, a lot of sort of extra support there in those cells. We divide them into 2 types, fibers and sclerids. The fiber plants down here are some pictures of some of those fiber plants. Long, slender fibers grouped in bundles. They can be more rigid, like in hemp. Sisal, like our baler twine we use is sisal, that they can actually take and twist and make ropes and stuff out of, all the way up to like cotton. Here's cotton or flax, that would have a much softer sort of fiber that we would use, you know, for our clothes and so forth. Sclerids are a little bit more varied in shape. A lot of times you'll see them almost with this star or kind of spiny shape. They give stuff more support, make stuff really hard. Seed coats, nutshells when you're cracking peanuts, walnuts, that kind of hickory nuts, that kind of thing. A lot of sclerids to give that really rigid structure. The stones inside of a peach, okay, those stones have a lot of sclerid fibers. Also pears, when you bite into a pear and it has kind of that gritty texture, that's because of the Sclerenchyma fibers inside that ground tissue of the pear, of the fruit of the pear. And finally, plant tissue number 3. So we had epidermis, we had ground tissue. Number 3 is going to be vascular tissue. Just like vascular, we think of veins, arteries, vessels that transport the blood or lymph through our body. Veins and vessels are going to transport 2 things. Well, there's 2 types. First type is xylem. Xylem is going to transport water and minerals. Most of the time we're talking from the roots to the leaves, okay, because where is water being absorbed in the plant? In the roots, and then it's taken up through the xylem, up to the leaves where it's needed for photosynthesis and to replace the water that's being evaporated, that's being lost out of the stomata, the little openings in the leaves. Xylem, there are 2 types of xylem. The vessel elements, these right here are the vessel elements. They are larger. They form almost like a pipe, and some of them at the end have a perforated plate, okay, that allows the water to move up through. Okay, usually alongside of them is the second type called the tracheids. The tracheids have more of a tapered end, like this, instead of just being sort of a round tube. The tracheids have more of pointed ends. They're a little bit slimmer, and water moves kind of across them and side to side, and they have these little pitted walls where the water would move up through and then across and then continue on its way. So over here it would come up across the cell membrane and then continue up the plant. Oftentimes they are side by side. If you look at the picture over here, here you see the vessel, and then over here you see the tracheids over here, with, for support, some parenchyma cells in between, just lending support to that vascular tissue. So remember, xylem, you got vessels and tracheids, and they are important for water and mineral uptake. And finally, vascular tissue, second one is phloem. Phloem is important for sugars and sometimes other organic molecules that are produced out in the leaves. They're usually taken back to the roots for storage. Again, most of the time from leaves to roots, although we do see it here in Ohio. In the springtime, you see people tapping maple trees as they come out of their winter dormancy. The sugars are reversed, the flow is reversed, and the sugars come back up out into the stems, out into the branches to provide sugars, to provide energies for leaf production in the spring. So we drill in, we tap, and we get some of that phloem. We boil it down and make that yummy, yummy maple syrup. Okay, but most of the times in the summer, once the leaves are formed and they start photosynthesizing, okay, the flow goes from leaves to roots back so that there's storage. There's two types of phloem cells as well, the sieve tube members and also the companion cells. Sieve tube members are more like the vessel elements would be in the xylem. They form more of a tube or a pipe-like structure. Okay, so over here would be the sieve tube. This would be this part right here, okay, the thicker one. They are like a sieve plate. They have a sieve plate, kind of a plate with holes in the end that allows the sugars to move through it and kind of form one continuous pipeline. The companion cells, the companion cell would be this thing here kind of stuck on the outside of it. Over here you see a companion cell kind of against the sieve tubes over here. The companion cells do have a nucleus. Okay, you can see the little dot in there. They are oftentimes important in maintaining the sieve tube members. And there's connections there, again, if we remember about our plant cell to cell connections called plasmodesmata. And their job is to sort of maintain the sieve tube members. And here's kind of a picture of that happening as well. So again, look at, here's some of our ground tissue, parenchyma, that's adding some support here. And then your sieve tube member here, the big pipe, and then your companion cells here alongside of it that are just providing nutrients and support to maintain those sieve tube members. Okay, and that's it for the first section of chapter 24. In the next section, we'll begin to look at some of the differences between monocots and dicots.